Thank you very much, Scott. So you may have seen that I've undertaken a wardrobe change, and so we're pulling all the stops like in a Beyonce or Taylor Swift concert. And this is to thank Mark Blaxter, our next speaker. He was very kind to bring a shirt with him that celebrates one of the accomplishments of the Tree of Life program. And the Sanger Institute is our largest user in the plant animal space. We've been cherishing the partnership for probably 15 years now, and I'm very honored and excited to have Mark talk about the latest programs that he's leading them.
Yeah, so hello everyone. It's a great pleasure to be here. I'm Mark Blaxter. I've got the honour of leading the Tree of Life program at the Wellcome Sanger Institute. At the Wellcome Sanger Institute. So the Wellcome Sanger Institute started out by sequencing genomes, took a detour into doing things like resequencing genomes and doing single cell stuff and all sorts of other things, which wasn't sequencing genomes. And more recently, over the last 5 years, we've returned to our roots and we've been sequencing genomes at scale. The first genomes the Sanger sequenced were biodiverse genomes. There was a nematode and a human, and then a few other things. But now we really are sequencing genomes at large scale. So we have a series of projects which are running at the Sanger Institute in our program, which is called Tree of Life. And these projects are all focused on sequencing reference quality genomes, really high quality reference quality genomes for different bits of biodiversity. So our major one is one we call the Darwin Tree of Life project. This is sequencing all the organisms, fungi, plants, protists, and animals that live on the land and in the seas around the islands off the west coast of Europe, which are currently occupied by the nations of Britain and Ireland. So this is the Darwin project. There are about 73,000 species we share those islands with, and we aim to sequence as many of those as possible. So that's our biggest project. But we also work on several other projects, including projects which are using host and symbionts at the same time. We have projects that extend beyond these islands off the west coast of Europe to the whole of Europe. So Project Psyche, we hope to sequence all the Lepidoptera of Europe. So that's about 11,000 species. That's for climate change monitoring, that's for pollination support, that's for looking at pest organisms, and also for looking at evolution, because butterflies evolve really fast and are very pretty. And then we also collaborate with the Vertebrate Genomes Project, the European Reference Genome Atlas. And all our work is done under, if you like, the umbrella of the Earth Biogenome Project, which aims to sequence all life for the future of life. So I'm going to show you a little bit about how we're doing this. Obviously, I wouldn't be here if PacBio wasn't at the heart of what we do. And then give you some little surprises that have come up in recent times, or interesting things that have come up in recent times in the genomes we've been sequencing. We are interested in the science of these genomes rather than just producing more and more and more, although we are very proud of how many we produce and the quality. And so I'm not going to give you any deep scientific stories about what we're doing, but I'm going to indicate some of the interesting things that have come up that we observe in these genomes that tell us something about new biology. We've built what we call the genome engine, and our snake of the pipeline goes down and up and down rather than left to right, but it's the same sort of pipeline as you saw for the ocean genomes. We find specimens, we get them identified by our biodiversity colleagues, the taxonomists and naturalists who know what they are. We get them in, we extract them, which are extracting from a vast range of different taxa, and so we have a vast range of different extraction methods to get the best possible DNA out of these, and then we sequence them. So we use high-C long-range and mostly PacBio long-read sequencing to build the genomes, and we also do RNA-seq for transcriptomes, not to do any functional biology, but just for gene finding. Generate a lot of data. Those data are then go through a primary assembly, scaffolding, detailed curation to make sure they're correct. All our assemblers and scaffolders have a ground truth on a set of genomes, humans mostly, and therefore they make category errors when they're assembling other genomes. There are also issues in other genomes which confuse the assemblers and scaffolders, and therefore human intervention to make sure those genomes are absolutely correct is essential. We then submit all our data publicly to ENA, and from there it goes into the INSDC source into GenBank. It's really important to us that all our data are public as soon as is possible. Ensemble does our gene finding and feature annotation, data presented through an open portal. We publish a genome note, and then those genomes are free and available to anybody to use. So just to point out where PacBio data comes in, on which side is that? Anyway, on the left-hand side, is it? PacBio data comes in on the left-hand side and goes through the genome engine assembly pipeline to generate very high quality genomes. I'm not going to go into this in any detail because you've seen this. We do a lot of PacBio sequencing. So this graph shows the yield on the y-axis. Since we started the Tree of Life program at the Wellcome Sanger Institute in 2019, the red dots are obviously from our SQL instruments. They start at quite high yields, very quickly got into running HiFi data, switch to the SQL 2e, and then you see we swap over from SQL 2e to Revio, when they were released, and we're now only running Revio. Of course, all these dots are printed on top of each other. Those of you who are looking at this carefully will see that we haven't universally got higher yields on the Revio when we shifted to Revio, so it wasn't just we. There are still some samples which are poorly performing on Revio. That's usually because of DNA quality, as far as we think. Could be due to co-purifying contaminants, which are somehow interfering with some of the pieces of the biochemistry and molecular biology of generating PacBio libraries. But we are usually generating much more data on the Revio than we were on the sequels. And of course, we're looking forward to having that number go up from the 90-ish gigabases to 120 gigabases with the Spark chemistry. We put libraries from over 4,000 species on the PacBio since we started. You'll see it's approximately 50% arthropods. That's because there are 50% arthropod species in the world. There's nothing we can do about that. There are slightly more Lepidoptera than maybe there should be. We have a certain enthusiasm for Lepidoptera, partly because there's a lot of DNA in a juicy moth or a butterfly, but also the genome's sequenced beautifully and assembled beautifully, and well, the organisms are beautiful. That's about 300 terabases of raw data and 1.8 terabases of assembly. As I say, this is across the tree of life. So we've sequenced protists, so single-celled organisms from a wide range of protozoan phyla, plants, including single-celled algae, but also mosses and higher plants, many fungi, including yeast, but also mycelial fungi. Then the animals, everything from the jellies, the comb jellies and the jellyfish, lots of deuterostomes, lots of vertebrates, lots of spiralians, mollusks, nemerteans, annelids, and then, as I said, many arthropods. So this tree is a taxonomic tree rather than phylogeny, so it's just showing the relationship to the organisms in the taxonomy, and the red histogram shows the number of species we sequenced. You notice that out to the left, where the butterfly is, we have sequenced rather a lot of butterflies and moths. In October last year, we hit an amazing milestone, which is sequencing 2,000 genomes. So there are many more than 2,000 genomes in the IMSBC databases. There are thousands of genome sequences. The point about these genomes is that they are high-quality genomes. So they meet the EBP metric. So the EBP metric is a series of scores which the EBP has proposed that we attempt to reach in order to really get at the biology of the genomes underneath, so we're not missing anything categorically from the genomes. So all our genomes are 6CQ40. So 6 is the 10 to the 6 bases, at least, a megabase, counting N50. C means chromosomally complete scaffolds, and Q40 is the QV value of the bases in the assembly. So that's what we aim for. So they are not just 2,000 genomes, they're the best possible genomes we can make. And it turns out that the 2,000th, we submit genomes in batches, and the batches go into the database in one go, and then they come out random, in a random order afterwards. So we're not exactly sure what was the 2,000th genome, but we have decided or declared that the 2,000th genome was this one. It was part of the batch that went in for 2,000. So it was seen as part of the Darwin Tree of Life project. So it's British and Irish species. And it's water milfoil, so it's a plant. It's got a relatively small genome, so only 245 megabases. It's actually an important genome for America as well, for the US as well, because water milfoil is an invasive species in this part of the world. And it blocks waterways and crowds out native plants and affects native freshwater invertebrate diversity. So this plant is not just for Darwin. We like this plant because it's a British plant or an Irish plant. We like this plant because it's part of our native biota. You like this genome as well in the States because you can possibly use this genome to think about ways of eradicating it. And it's very odd. We think about sequencing these genomes for conservation, but sometimes it's a two-edged sword. We can sequence the genome so we can conserve it in places where it should be, but the genome can also be used to eradicate it. We sequenced a very large genome, the genome of a newt. This is a triton, and this is a 40 gigabase genome, 27 gigabase genome, and it's really exciting to have that for people conserving newts in Britain and Ireland and across Europe. But it's also quite exciting for people in Australia, especially southern Australia, where the newt is an invasive species and is eating native water organisms. And so they're really interested in the genome because they want to work out how to kill it. So they're going to use this genome to investigate things like interfering with the reproductive biology of the newt in southern Australia so that they can remove it from the ecosystem. We work with lots of people and we want to give them credit. And one of the ways we give them credit is by publishing every genome as a separate paper. So this does mean that our project has published well over 1,000 papers since we started, which sounds really good. Each one is a genome note, a data note which tells you exactly how good the genome is. They're citable, and the important thing for us is that the first author is the person who collected the organisms. It's often the person who's lost in multi-genome publications. They're all published openly at Open Research, and those publications are also, if you like, partially interactive because they link back to various displays and representation of the genome in other databases. We want to see projects like ours elsewhere in the world, so we make all our protocols and pipelines available. So for example, our computational pipelines are built as Nextflow pipelines and are available through our pipelines website here, but we've got open access to all our data. So our wet lab protocols go out to protocols.io. Our data is available in various project-specific portals. We actually let you look at our raw data. We have a QC site where you can actually go look at the quality of the data as it comes off the machines. You can see what data was produced yesterday. I haven't looked yet, so you'll find out before I do what we sequenced yesterday. All our other data are available publicly. So, I'm just going to quickly go through some recent entertainments, some interesting genomes, and interesting biology we found in genomes we sequenced. So we were very proud, a couple of years ago now, to sequence the genome of Mistletoe. So Mistletoe is a hemiparasitic plant. So it's green, it photosynthesizes, but actually gets most of its nutrient from the host trees on which it lives. So it's an interesting organism in that way, but it has a stupidly large genome. So the genome is 30 times the human genome. It only has 10 chromosomes, so each one of those chromosomes is 3 human genomes, end to end. And this assembled.
Oh, there you go.
Okay. So it took a while on the machine to assemble, but it did assemble. It's not that it fell over and died, which you might have thought it would. It is full of repeats. So usually you'd expect a genome like this not to assemble because it's so full of repeats. Big genomes, more repeats, more chances for your assembly graph to go horribly wrong, failed assembly. That's not what happened because it turns out that genome biology of organisms with very big genomes is very different. And repeats accumulate mutations so they drift away from each other. And so the repeats are actually different from each other. They're not purged like they would be in an organism with smaller genomes. And therefore, they're sequence unique, and therefore with one PacBio reads, you assemble through them. So the contig N50 is 33 megabases. The scaffold N50 is 3 human genomes. The thing I find really interesting, this is a female we sequence. Females are XX, males are XY. So no, females aren't XX. Females have 5X chromosomes. So 5 of these chromosomes, probably the 5 smallest ones, karyotypes, are X chromosomes, which are therefore haplosufficient in males. So somehow this organism has managed to do dosage compensation across something like 40 gigabases of sequence. Yeah, so I don't know how it's done it, but it's done it. So it's XY is actually 5X, 5Y species. So we resequenced a male, so we have an idea which the X chromosomes are. We haven't tried to assemble that male because this took, this was sequenced on the sequel, this took over 100 sequel runs. So these HiFi plots are beautiful. So this is the same pattern as you saw previously. Scott's talk using the HiFi data to do joint curation of both haplotypes. And it's very, very powerful. And the second one here, the Urtica diurca, the common nettle, is a tetraploid. And again, you can see here in the inset that's blown up there that Joe and his team are curating all 4 haplotypes of the assembly. So one that came up recently, which plants are wonderful. This is a dog rose, Gertrude Stein, famous American poet. A rose is a rose is a rose is a rose. She should have added another is a rose at the end of that. Because if you look at this HiFi plot here, there are 5 chromosomes, or 5 segments, in each of these parts of the HiFi. So here's just one chromosome pictured random. There are actually 5 copies of each chromosome. It's pentaploid. 2 of those copies recombine and do meiosis. We think that's probably the 2 copies at the bottom of this segment. The other 3 copies are passed on parthenogenetically, so without recombination. Again, because actually the HiFi reads and the drift, the divergence between these copies is possible to assemble all 5 independently. So I didn't know that roses were pentaploid, the dog roses were pentaploid. Many, you can imagine many show roses and garden roses, because we pick them for showy flowers. Talks on roses here. You get tetraploid roses in a cultivated rose. This is the wild rose, dog rose, which is pentaploid. So chromosomes. Why have we got 23 chromosomes? Well, because mum and dad did. But in the 5XX, why have we ended up with 23? What's driving the number of chromosomes we have? It's actually something to do with the number of centromeres we have. So number of centromeres is actually what drives that. What happens when you don't have centromeres, when you're holocentric? So Lepidoptera, butterflies and moths, are holocentric organisms. They don't have specified centromeres in their chromosomes. And you'd think that would allow butterflies and moths to have crazy variation in their chromosome number, because a broken chromosome could still quite easily segregate properly in division. In fact, most butterflies and moths have 31 chromosomes. So there's something absolutely constrained about butterfly moth chromosome evolution. This is a graph of chromosome counts across 500 species of Lepidoptera. That said, some Lepidoptera do have crazy chromosome counts. So this little butterfly here is the Atlantic blue butterfly, which flutters around in the mountains of Morocco. Very pretty little thing. It's probably endangered. It's quite rare. And it is the organism on the planet with the record number of chromosomes. So, Lukatov counted meiotic plates and counted 220, at least 226 chromosomes. You can see here there's 2 large chromosomes in the middle there, and then lots and lots of tiny little chromosomes. He's actually put a dot of red ink on each one here, because he was counting them. So this is the organism with the highest known karyotype of any diploid organisms. There are polyploids with many more chromosomes than this. So we thought we've got to sequence this. Okay, so we found one specimen which had been caught way before the Tree of Life project started. and he'd been living in a freezer in Spain for a while, and then lived in a freezer at Oxford for a while. And so a PhD student went across to Oxford and collected it and took it back, and it sequenced both HiC and PacBio sequence. And we've assembled it. And actually, the one we sequenced, the female, is now the record holder, the organism on the planet with the highest known chromosome number by non-polyploid eukaryote. Has 227 chromosomes. The genome, it's not polyploid. The genome is only slightly larger than its relatives, which have many, many fewer chromosomes. 224 of those are tiny, an average of about 2.2 megabases. So tiny chromosomes. And then it has 3 large chromosomes. Those 3 large chromosomes are sex chromosomes. So it has 2 very large Zs, which are derived from the ancestral sex chromosome fused to an autosome, and another autosome which is taken on the life of a new Z, and it has a W chromosome which has bits of those autosomes degenerated away. So the W, so these are ZW heterogeneous female species. So this tells us 2 things. One, probably 2 megabases is the smallest a chromosome can be in butterflies and moths, and it tells us something about the rules, and we can look at what's driven and what the precursors to driving the chromosome to be this small. It's something to do with chromosome structure, I can tell you. And the other one is that the sex chromosomes are insulated from whatever it is that's chopping the genome up. So whenever these breaks are happening in these chromosomes, it's not happening to the sex chromosomes. So there's something special about being sex chromosome. So this is the worst, I think, in terms of chromosome count we're ever going to get in a diploid. I'll do this a bit quickly because it's mentioned. We're really interested in lowering the sample input requirement barriers. If you've got an elephant which weighs 6 tons, there's a lot of DNA in an elephant. I know it's not very legal to sample an elephant, but there are many tens of kilos of DNA there. The smallest land vertebrate is a very small frog, it's about half a gram. There's still enough DNA in that half-gram organism to do a reasonable genome estimate. But it's not actually just a piece of lint on the screen there. That's actually a nematode. Nematodes are tiny, they have 1,000 cells. The maximum amount of DNA you're going to get in those 1,000 cells is something like 200 picograms. So we have to have a method that allows you to do genomes from nematodes. Who cares about nematodes? Well, I do, but we all should. They're actually the most abundant animal on the planet, not in terms of species, but in terms of species numbers. And every one of you probably has been infected by a nematode. All our farm animals are infected by nematodes. All our farm crops are infected by nematodes. So they're major parts of the ecosystem. So we have this method called PIMS, which was developed by Chris Lawmer. It's the basics of the process is that you extract the DNA and you shear it without measuring its mass or anything. And then you amplify that sheared DNA in a controlled way such that you generate enough DNA to sequence. We can extract the RNA at the same time, so we can also make cDNA for isoseq. And this is an example for this nematode, Odontocalamus thalassophagus. The PacBio came from one nematode. The HiFi, we still haven't got to work on one nematode reliably, so the HiFi here comes from 22 of these species nematodes. And we have the first time for a whole class of nematodes, we have a chromosomal genome assembly. The other thing, as also mentioned here, microbiomes are a thing. We're seeing lots of organisms pulled from the wild, and it's really common to co-sequence another organism along with it. We call them cobiotes because we don't know if they're pathogens, parasites, microbiome, accidents. And so, for example, this moth, Bucephala, moth-tip moth, has beautiful 31 nuclear chromosomes, a mitochondrion, and 3 genomes of about a megabase of 3 different strains of Wolbachia. So Wolbachia is an alpha proteobacterium that lives inside many, many insects. And this one just happens to have 3 different strains, which come from 2 different supergroups, so supergroup A and supergroup B. So not just Wolbachia we find. Here's another Lepidopteran, which has a Wolbachia. This is a high C plot. But it also has a microsporidium. So microsporidia are very small genomed eukaryotic parasites. So these are the smallest known genomes of any eukaryote. They go down to about 2 megabases for a genome of a eukaryote. This one is slightly larger than that, but we can now assemble also the eukaryotic parasite that lives inside this moth, which was carrying 2 probiotics. This goes to a crazy scale. In our aquatic symbiosis project, we sequence a lot of sponges. Sponges suck things up from the environment, but also many sponges have lots and lots of endobacteria, bacteria living inside their cell. And so in our aquatic symbiosis project, we sequence a lot of sponges and other things with microbial symbionts. This is a tree, again a taxonomic tree, of the microbes, the prokaryotes. The grey segment next to the scale is the archaea, and then lots of bacteria. And the red dashes show you where they were found in a series of 300, in a series of 160 symbiotic systems. In total, we have 5,500 metagenome assembled genomes, which are essentially circles. So we've got another gigabases of data from those. Do you want me to start? Okay, I'll skip through this, but I was going to show you how PacBio assembles beautifully over repeat sequences and real bits of genomes that otherwise wouldn't be seen. I'm just going to thank every tree wife at Sanger. It's an absolute pleasure to work at Sanger with the team that I work with. And they're not only experts, but they're enthusiasts. And they do amazing things with amazing species. We collaborate widely, and I really thank all the people who bring specimens to us. They don't have to do this in most cases, but people really like getting their favorite sequence, species sequence. And then we work very closely with the long green team, Sanger operations, to fill these PacBios. And those are the people who are